All right, this video is going to introduce some of our processes that make up our second line of defense. So we're still talking about our nonspecific immune response, also called our innate immune response. And so the second line of defense is really going to come into play once pathogens have already gotten inside the body, okay? And so these are going to be more present, deeper within the tissues, okay? A lot of our first line of defense, they had roles at body surfaces. So now we're thinking about, well, what happens once pathogens get past our first line of defense? They've gotten into our bodies, they've perhaps attached to cells or gotten deeper into tissues. Well, now what? What are we going to do to try and eliminate those pathogens so that they can't continue to replicate and make it cause an infection and make us sick? So here are, 7 different processes. We're technically going to talk about an 8th as well, but it's going to be like a very short, like 30-second spiel on antimicrobial peptides, because just not much is known about them. So we know they exist, we have an idea of what they do, but we don't know a whole lot else. Okay, so I didn't include that on the list because the bulk of the assessment items for this content is going to focus on these 7 processes. So let's go ahead and jump in, and we'll probably knock out interferons and phagocytosis in this video. Okay, so we're now really getting into immunology. Okay, so thinking about how our immune system kind of acts as like a surveillance system of the body, kind of constantly on the lookout for pathogens and signs of invaders. And then they have to be able to recognize self from non-self, right? We don't want our immune system attacking our own healthy, uninfected cells, but it does need to be able to recognize a foreign pathogen or an infected self cell, okay? So white blood cells are going to be what we talk about here for our immune cells, and they are constantly moving throughout the body. They're on the lookout for pathogens, okay? And once they recognize pathogens or infected cells, then certain processes are going to kick into gear, and that's going to be what we talk more about right now. Okay, so let me kind of explain a little bit more this self versus non-self, because it's going to play a role in our acquired immunity as well. So look at the purple cell on the far left. Sorry, I got ahead of myself with the animation here. Let me back up a little bit to help you focus. So we're going to have our white blood cell, okay, and it's one of our immune cells. And so it is going to have receptors on it that are going to look for specific molecules on the surface of cells that it comes into contact with. So as you can see, the orangey-ish cells with the blue molecules on it, that's a normal, healthy, uninfected self cell. That is a cell that belongs in our body. It's ours, okay? We want it to remain there. And so you'll notice when the white blood cell comes into contact with them, it binds to it and assesses it. It's like, oh, yep, you have the receptor I'm looking for. You belong here. Great. Go on about your business, right? You see nothing happens. There's no reaction. But when the white blood cell comes into contact with a pathogen, okay, it is going to recognize, right, those little molecules on the surface. We call those PAMPs. Pathogen-associated molecular pattern. You don't have to remember what it stands for, but when you see PAMP, then you need to know that's a pathogen we're talking about. It's part of the pathogen that our immune system recognizes. So the white blood cell binds to it, and they say, wait a minute, that's not what I'm hoping to see. You're a pathogen. You need to be destroyed. So some white blood cells are phagocytic and can go ahead and just ingest and digest it. Other white blood cells might detect it and then kind of release signaling molecules that say, woo, woo, hey guys, we're infected, there's a problem, let's get rid of this, okay? So just so you know, not all white blood cells are phagocytes. But the point of this image is to show that once a pathogen is detected or an infected self cell is detected, then it's targeted for destruction one way or another. All right, so interferons are really cool molecules, molecules that infected cells tend to make. And there are a variety of interferons that exist that will kind of alert the body to different types of infection, okay? But in this example, we're gonna use a virally infected cell, okay? So essentially what happens is when a cell becomes infected, for instance, by a virus, that infected cell starts to produce molecules called interferons. And they are meant to interfere with viral replication and the spreading of the virus to neighboring uninfected cells. Okay, so what you might want to jot down is the cell on the left is an infected cell. Only infected cells produce interferons. How I think about interferons is I think of them as like an alarm system. So pretend everybody right on your street has an alarm system. You go to bed at night, you set the alarm, and unfortunately your house is targeted and burglarized, right? So in this example, you're quote unquote being infected. We're going to pretend that the intruder is a virus. So once the intruder gets in, your alarm system goes off. And what does that do? It's alerting all those uninfected, unburglarized houses and people inside of them that there's a problem. There's an intruder in the vicinity and we need to prepare, right? We need to call 911 or whatever. And so the interferon is the alarm system. It's a message being sent out to neighboring uninfected cells that there is a problem and they need to prepare for a potential problem. Okay, and so interferons have been shown to kind of trigger multiple responses. So for instance, so on the right in this image are kind of different scenarios, different roles that interferons play. So interferons signal to neighboring uninfected healthy cells to start eliminating RNA that's present in the cell and to reduce protein synthesis. Because remember, some pathogens like viruses require host resources in order to live. So by destroying RNA, they're getting rid of some of those proteins and enzymes that the virus would need in order to be successful. So it's kind of stopping or preventing viral RNA replication from being able to occur. Interferons can also act as signals to neighboring infected cells to undergo apoptosis, which is programmed cell death. It's essentially triggering that cell to die before the virus has completed its life cycle. Because if that cell dies before the viruses exit the cell, then the viruses die along with them, right? Because if the host is dead, they can't use the host anymore. So it's essentially like they're trying to just eliminate one cell to prevent the spread of the virus to many other cells. Interferons also activate immune cells, saying, hey guys, we have a problem over here, and they're going to kind of round up some of the big dogs to come and help eliminate the pathogen. Something that I would like you to write down for this section, please, as well. Interferons also stimulate uninfected cells to produce proteins called AVP, for antiviral proteins. And so these little AVPs are made by the uninfected cell as a way to prepare themselves for a potential viral attack. And so I kind of envision the AVPs as like a little army at the border. They're like, all right, virus, we know you're out there. You may try to come get us, but we're ready. And so then if a virus does try to infect that cell, those AVPs can eliminate it before it's able to actually initiate and complete a replication cycle. Okay, so that's two things that the interferons are doing for uninfected cells. But these are really snazzy. So again, just kind of big picture snapshot. infected cells make interferons, which are meant to interfere with the pathogen's replication cycle to prevent the spread of that pathogen. Okay, so the interferons are made by the infected cell to warn uninfected cells or to trigger infected cells for apoptosis, to shut down the spread of the pathogen that way. All right, let's go ahead and end this video with a quick talk about phagocytes and phagocytosis, one of my favorite processes. So phagocytes are going to be surveying various parts of the body and hunt down any pathogens or other things like injured or dead cells. They ingest them and digest them, okay? And we'll talk more about that third bullet there next week when we talk about our acquired immune response. But phagocytes have a role in both innate immunity and acquired immunity. Okay, so 3 different types of phagocytic cells you're going to hear about over the next week or so are going to be neutrophils, eosinophils, and macrophages. Okay, and you can see kind of what their roles are here, but we are going to come back to them in other videos. All right, so phagocytosis, there's a lot on this slide, so I want to kind of talk through it a little bit and kind of give some extra detail. So a phagocytic cell initially is on the hunt. It's on the prowl for pathogens. And one way it detects them is because, for instance, with bacterial cells, as they're going about their business, right, trying to survive inside of our bodies, they naturally just release little chemical byproducts that serve as signals for the phagocyte, okay? So it's kind of just convenient, right? The bacteria is not trying to be found, but molecules that it's naturally releasing as byproducts are able to be detected by phagocytes. So I think of step 1 as like in movies when somebody's gone missing, and so they give a hound dog, you know, somebody's t-shirt so they can sniff it and then they go chase the scent. That's essentially what chemotaxis is. The phagocyte is quote unquote sniffing out the chemicals left by the pathogen, and it becomes a game of like chase. Okay, they hunt them down. So once the phagocyte has chased down the pathogen, it has to attach to it. Okay, just like in a game of tag, right? If you're chasing after somebody, once you're close enough, you reach out and you grab them, right? And so in this case, that contact, we call it adherence, they're attaching. And one of those PAMPs, those pathogen molecules, is going to bind to a very specific receptor on the phagocyte called a TLR, a toll-like receptor. Okay, so that is how the phagocyte contacts and grabs hold of the pathogen. Okay, so the PAMP on the bacterial cell, for instance, is going to attach to a toll-like receptor on the phagocyte. So at this point, the phagocyte is like, aha, I gotcha. And so now it needs to actually engulf it, okay? And so you can see on this picture where there's multiple pathogens, right, attached to the phagocyte. And you see how it kind of looks like the phagocyte is coming in for a hug. So what's happening is the phagocyte is kind of reaching it's kind of creating fake-like arms, in my example, to come up and enclose the pathogen. Okay, so those little extensions that look like it's hugging the pathogen are called pseudopods. And so those pseudopods kind of keep extending out until they touch and bring in the pathogens into a little vacuole or a vesicle. Okay, so now the pathogen is inside the phagocytic cell. It has been ingested. So that little vesicle or vacuole there that contains intact living pathogen is called a phagosome. And what's interesting is these phagosomes have proton pumps, which is pumping protons into the phagosome. And you might be thinking, why do we care, Chelsea? Why is that? Well, right now, these pathogens are alive and well. We don't want that. We want to digest them and eliminate them. Well, which eukaryotic organelle is responsible for digesting material? Well, that would be my favorite, the lysosome. And remember, the lysosome has those hydrolytic enzymes that require an acidic pH. Now look at this picture, and while it's not entirely to scale, you can see that those incoming lysosomes are quite a bit smaller than that phagosome. And so we need that phagosome to, the pH of that phagosome to be pretty low when the lysosome fuses with it. That way those hydrolytic enzymes will work. Okay, so there are proton pumps in the phagosome trying to decrease that pH, because notice in step, well, my list is step 4, it's 5 on the picture, which is fine. Basically, the lysosome is going to fuse with the phagosome. So it's depositing all those hydrolytic enzymes into that vacuole, okay? And so we call this new vacuole the phagolysosome, because it still has the original pathogens, but now the hydrolytic enzymes have been released. And so next is going to be digestion, aka microbial death. And those hydrolytic enzymes are going to destroy the pathogen, okay, killing them. So then once that process has ended, now the vacuole contains just residual undigestible material. And so now we refer to that vacuole as the residual body. And that residual body is going to exit the cell via exocytosis to release all that undigestible debris, because it's no good to the cell. Okay, so again, kind of a quick picture rundown. The phagocyte is going to chase down the pathogen via phagocytosis. It's going to grab hold of it by having its toll-like receptors attached to the PAMP on the pathogen. Then the phagocyte is going to form pseudopods to kind of wrap around and engulf those pathogens. That leads to the formation of the phagosome. The phagosome is going to have proton pumps so that it's gradually decreasing the pH. So when the lysosome comes and fuses with the phagosome, which we now call the phagolysosome, those hydrolytic enzymes will be active because there'll be a low pH, and it will chop up and digest those pathogens, killing them. So once that process is done, now all that's left is undigestible cellular debris, which the cell doesn't need. So this residual body, which is what we now call that vacuole, is going to leave the cell via exocytosis. All right, y'all, that is going to be the end of this video. So let me know if you have any questions.